I want to just really briefly go through the exam format. And Pete, can you see me okay? All right. So the exam is happening on Monday of this week. So I just want to make it really clear. On Monday, we're having an exam. This is all in the syllabus, but I think it's nice to just repeat it. There's no formal class meeting. Instead, you can use the time to be working on the exam. I estimate that a student who has studied and has prepared ahead of time will take somewhere between 70 and 80 minutes to do the exam. I'm designing the exam to be very equivalent to one that I would offer if we were not in a remote learning kind of setting. It's open book, it's open note, and it is open internet, right? So really you can use anything at your disposal except for a friend, a family member, or a faculty member at the college. If you have questions about a question like what it's asking, feel free to ask me. The worst thing I'll say is just do your best. I can't answer that right now. Okay? And so the format of the exam is I'm going to post it as a link to a Google document on Moodle. So to find the exam, you'll go to Moodle, you'll go to next week's activities, you'll see something that says link to the exam, and you'll click on it, and you'll get to a Google document that's read-only. No one can edit it. Just make a copy of it, change the name to your last name plus exam. So I would change it to Hovey Exam 1, Biology, something like that. And then when you're done, and then save it in your folder that you've already shared with me where your other labs and things are. And then when you're done with it, put a link. Let's see, so I'll just, you'll save it to your Google folder, and then to turn it in, you'll submit it via the Moodle course webpage. There will be an assignment submission portal, and it'll say make a link or provide a link or something like that there. Any questions about that? There were just a few little kind of bumps along the road to submitting some of the labs, and I think that's super normal. If it happened to you, you're not the only one by any stretch of the imagination. But for the exam, I think the lab gave us a chance to make sure we know how to submit assignments. For the exam, I think you should know how to do that. The format of the exam will be a little different from the quizzes. I ask a lot of questions that might ask you to describe or explain something. I might ask you to draw something, like on a piece of paper, and take a picture of it and then paste it into your Google Doc. Because I know a lot of our students are interested in careers in graduate work and medical school or veterinary school, I'm not a huge fan of multiple choice, but I think it's important for students to get experience kind of working with that format. So I always have a few multiple choice questions on the exam. It's worth 50 points, which is not a lot compared to some of the other work that we're doing in class, but it's there to kind of have us sort of test our knowledge. Y'all seem so relaxed and chilled out. Am I reading the room incorrectly? Okay. My hope is that it's going to be kind of intellectually interesting and fun. We've worked really hard this semester, and so when you have the chance to kind of show what you know, that's also another way to sort of celebrate our learning, if you will. Okay, so why don't we take a quick moment to get your microscopes and to grab those slides. I set up lab today so that we can still sort of physically distance even though there's a smaller number of us here. I think one of the fundamental tools in the biologist's toolbox, right? I think it's not an exaggeration to say that our understanding of life dramatically changed when people saw the number of things that were moving around in individual little cells. And so For lab today, I have a written description of the different parts, but I want to just give you practically an opportunity to check out the microscope. So, if we look right here, these little guys, you can see they're kind of adjustable to the shape of your head. These are called ocular lenses, and the ocular lens it magnifies things to 10 times life size. Underneath, do you see right here, there's this sort of circle that has little lenses coming off of it. You can turn that either clockwise or counterclockwise. But I'm going to have you turn it counterclockwise now until you get to the smallest of those lenses with the red line on it. These are called objective lenses. And there's a 4x, a 10x, a 40x, and a 100x. There's 4 of them. The 100x is something called an oil immersion lens. Basically, we look at stuff by putting mineral oil in it. The refractive properties of water do not really interact well with super high magnification. That's why we use oil. We're not going to use oil immersion in this particular class. We're going to stick to these top 3 right here. But the combined effects of both of these lenses have a multiplicative effect. So in other words, the total magnification here for this little tiny nub with the red line on it would be 10 times 4 or 40. The intermediate lens is 100, and the blue lens is 400 times life size. And to see bacteria, we really, really want to be able to look at them at that 400x because they're so, so tiny. So remember, like 10 microns per cell. And although it's very tempting to want to just sort of start at 400x, a good technician is going to actually start at the lower field of magnification. That lets you find what you're trying to kind of identify. So let's get started with that. This part right here, the black part, is called the stage. It's flat and rectangular like a stage. We have slide clips that move in and out, so you can sort of fit your slide in almost like a little safety belt. So Fred, if you put yours in, there you go. And then the corner of it should kind of fit in any one you want. And then to the right of the stage and beneath it are 2 knobs. These knobs go adjust the stage so it can go vertically in your field of view, and then the lower one goes horizontally. Back in the olden days when I was taking biology, we didn't have microscopes like this. We had to actually move them around with our hands, and it was such a pain because one tiny move with your hand feels like a kind of marathon in the world of a microbe, and you end up kind of moving things around. Underneath, in the center of the stage, is an area where light comes up called the condenser. So let's plug in our microscopes if you haven't done so already. And there should be an on switch on the right. My microscope, helpfully somebody put a sticker here that says bulb needs replacement, so mine's not going to turn on, but you can kind of get the deal. So you should see light coming up through that center of the stage. That's called the condenser. It takes light from beneath and kind of focuses it into one plane. I'm almost done with my little microscope note. You can adjust the intensity of the light with the knob that's beneath the on-off switch. And then I want to draw your attention to the 2 knobs that are kind of concentric within one another along the lower side of the microscope. The big one is called the coarse focus. You can use it to really make dramatic changes in adjustment. And so what I do is I'll look at my little, whatever I have under my microscope. I try to find it in my field of vision first. And then I use the fine focus knob, which is the smaller one in the center, to actually crystallize and sharpen my image up. And for purposes of kind of just COVID mediation, I have some safety glasses that are next door that I use when I look in students' microscopes. But in general, I don't really want to spend too much time with my face on your microscopes in general. And then once you find your item in the field of view, you can go up to the next level of magnification, which is 100. Make sure that it's still there and visible. And then you can go up after that to 400. And what I'd like to do now is just turn you loose for, let's come back together as a class in about 10 minutes to work through the first part of the lab. And once you feel really confident in your ability to identify and work with some of these pre-prepared slides, I want to talk a little bit about just the biology of protists, and then give you some tips on how to actually work with living and live specimens. So protists are organisms that cannot be readily classified as plants, animals, or fungi, but they are eukaryotes. And sometimes they're kind of affectionately called the garbage bucket kingdom or the junk drawer of the tree of life, because essentially they aren't really united by common ancestry the way a lot of other groups are, like the animals, like plants, like fungi. Instead, they all have the feature of really not being like an animal, not being like a plant. Some of them have cell walls, some of them don't. Some of them photosynthesize, some of them don't. And what I think of when I think of the protists, I think of them as a really nice way for us to explore what eukaryotes look like and to think about two different nutritional modes. One of these is that mode of photoautotrophy. The other is heterotrophy. So just kind of, maybe spitballing a little bit, what do you think might be some features that would help us identify a photoautotroph? Val? Yeah, they're green, exactly. Yeah, they have pigments, right? So they are, many of them are green, but some of them are brown. So like anyone who's been to the ocean has maybe seen kelp. That's an example of a protist. Some of them are red. If you've ever had sushi, the nori that surrounds the rice, that's actually a red algae, and it has photosynthetic pigments that are red. And just fun fact, red is better at capturing light deep undersea than green is. And so we often see this kind of stratification in marine systems especially. But for purposes of today, I just want to give you a quick crash course on how to actually visualize these guys under the microscope. And behind Val, I have some heterotrophic protists like amoeba, like something called paramecium. They have pretty large cells. Behind Grayson, I have some photosynthetic protists like Spirogyra, the one that has the corkscrew-shaped chloroplasts. And we can look at those under the microscopes. And I don't want to overlook the bacteria, but I think looking at them on prepared slides is, I don't know if it's the best use of our time this morning. I have some live photosynthetic bacteria. They're actually a lot easier to see because they're green and colorful. And they're actually, for whatever reason, I think just more abundant in the slides that you will make than they are on our prepared slides. But by now, I feel like everybody at least has an idea of fine focus versus coarse focus, how to move your stage around, and how to adjust the ocular lenses to meet the shape of your head. So over here, I have some equipment for you, and I'm going to distribute this across 3 tables. Some protists like to move with flagella or cilia, and they're really fast under the microscope. So to slow them down, this stuff is called protoslow. It's slightly viscous, so it gives them, they have to work a little harder in order to move, so they're a little bit slower. So I'm going to put one here for Fred and Val to share, and one here for Grayson and Otto and Emma to share. And then what you'll do, I have a few different pieces of equipment here that we can get together. One of them is just a microscope slide. Who here has used one of these before? Maybe in a class? Yeah. When I use them, essentially, I'm going to just show you where they are. I also have some cover slips to go with them. I have two kinds of slides for you to work with. One of them looks sort of like this. And it's called a depression slide. And depression slides have like a little kind of they're kind of bowl shaped, so you can look at things that are in a more three-dimensional way with them that may be moving around, and those can be kind of interesting. Especially, I'd recommend it if we have time to go outside and grab some water out of the Swannanoa River. And then I also have flat slides. These are real easy to work with. All we do is you have your slide right here. You're going to take a drop of your water that has living things on it in it. You're going to take potentially a drop of your Proto-Slo, and you're going to just put those there, smash a covered slip down on top of it, and then look at it under the microscope. I'd like each of you, when you grab stuff, to also get either a paper towel or one of those wipes that are on the table right there, and that's because sometimes water wants to leak out of your wet mount. That's what this is called, a wet mount. And I don't want the water to get onto your microscope, so you can just use it to sort of dab around the sides of your slide. And that's it. Each of these vials is labeled, and when you look at something, I want you to try to figure out whether it's heterotrophic or whether it's photoautotrophic, and whether it's a eukaryote and whether it's a prokaryote. So today we're going for kind of very broad trends in our tree of life. If you get really psyched on protists, there are other ways for you to explore them in other classes here. All right, have fun. I'll be here. I'd love to look at your things and see the things that you're able to photograph too.